Hello friends, as stated in the cell theory, every cell arises from pre-existing cells. This is achieved through the process of cell division. The life of all multicellular organisms starts from a single cell, that is a zygote. The growth and development of every living organism depends upon the cell division. So before cell division, the cell undergoes sequential changes are called cell cycle. See here, the cell when decide for cell division, then the RNA and protein synthesis gets start. Then cell undergoing the synthesis of DNA. Later on, the cell again synthesize the RNA and protein. So doubled material in this S phase get divided into two cells and the cell division complete. In a cell cycle, the G1 phase characterized by synthesis of RNA and protein, S characterized by duplication of genetic material means synthesis of DNA, and G2 phase again characterized by the formation of RNA and protein like the tubulin protein, spindle fiber formation protein, and then cell divide into two. So these all sequential changes in a single cell is called cell division. So in this lecture we are going to learning the cell cycle. You all are welcome in my channel College Botany, and you are studying the cell cycle with Yuvraj. Dear friends, the cell division is the process by which a cell divides into two or four daughter cells. A cell divides when it has grown to a certain maximum size. The daughter cells form after the division of a mother cell may grow in the size and divide again. This constitutes the cell cycle. The period between two successive divisions is called generation time. A series of changes or sequential events which occur in the life of dividing cell is called cell cycle. The cell cycle includes the period from beginning of the one cell division to the beginning of Next, cell division. The cell cycle consists of two main phases: interphase and M phase. The figure itself indicated the interphase and M phase. So interphase comprising the G1, S, G2 and G2, and M phase comprises the mitotic cell division or meiotic type of cell division. So the cell cycle shows two division, two phases, interphase and a M phase. The interphase was formerly called resting phase, but the cell is highly active and prepare itself for the ensuing division. Therefore, it is now called preparatory phase. It involves many metabolic activities. The interphase can be further divided into three phases: G1 phase, S phase and G2 phase. G1 phase, S phase and a G2 phase. So first of all, the G1 phase. The G1 phase is first growth phase. First growth phase. It is also called post-mitotic gap phase. It involves synthesis of proteins and energy molecules required for DNA replication. Proliferation of cell organelles takes place, or cell may withdraw from cell cycle and enter into G0 or quiescent stage. The cell in a G0 state may re-enter in G1 phase or may get differentiated and become a permanent cell. The loss of control at G0 lead to uncontrolled division as in the tumor or cancer cell. The second phase is S phase. It is also called the phase of synthesis or synthetic phase. The replication of DNA takes place during this stage. The entire genetic material, that is DNA, duplicates or synthesis. Nucleus become double sized. Histone proteins, which are required for organization of chromosomes, are also produced. The next phase is G2 phase. It is the second growth phase and also called pre-mitotic gap or growth phase. It involves the synthesis of tubulin protein, the spindle protein, and RNA. There is duplication of cell organelles such as mitochondria and chloroplast. The centrioles duplicates and two centromeres are formed. So centrioles duplicates and two centromeres, please correct it, two centromeres are formed. Thus, the cell prepare to enter in mitotic phase. The next phase is M phase, means cell going for cell division, that is prophase, metaphase, anaphase and telophase. Now we are going to learning the mechanism of cell cycle. In the cell cycle, both unicellular and multicellular organisms, for each event in the cell cycle, there is trigger or signal reactions. This signal controls the entire mechanism of cell cycle. The point at which signals are necessary are called checkpoints. The two important checkpoints are in G1 and at the end of G2 phase. During G1 signal, is released to enter the replication cycle to enter into the S phase, while at the end of G2 phase the signal is released to start the nuclear division. Due to presence of such checkpoints at certain stages, the reaction are arrested, and after obtaining the necessary signals, the reaction gets starts. The checkpoints in the cycle can inhibit transition from one regulatory state to the another until some internal or external conditions has been satisfied. So this is the G1 phase. These are the three checkpoints: pre-start G1 checkpoint, start G1, and post-start G1, and other one at the end of G2. We are studying here the example of yeast cell. So in yeast, the G1 phase in the cycle is divided into three substages, and each substage is a checkpoint. These three substages in the developing yeast cell are pre-start G1, start, and post-start G1. These three checkpoints control different aspects. The first checkpoint confirms the nutrient in the system are sufficient and there is no starvation. So pre-start point confirm whether the nutrition is there and there is no starvation. It also shows the cell is healthy and signal released by this checkpoint to start replication cycle. This signal is identified by second checkpoint, that is start point, and it start to synthesize the material required for that. So third checkpoint confirm that the cell has attained required size and maturity, then release the signal for S phase. The third checkpoint, that is post start G1 checkpoint, release the signal for the S phase to start the synthesis of DNA, replication of DNA. So all these signals get in the form of mating pheromorphs like alpha and beta. Most of checkpoints are present in the G1 phase for cell cycle regulation. In unicellular as well as multicellular organisms, There is a central cell cycle control system which regulates the progressive and ordered sequence of reactions during the cell cycle. So this is all about the cell cycle. Thank you. You all are requested to subscribe the channel and press the bell icon so as to you will get earlier notification regarding the next topic.